Well, I thank the organizing committee for having RevOpsis Therapeutics here today. RevOpsis is bringing the era of multispecifics to retina. We've heard a lot about multispecifics in oncology, and it's time for it to enter into retina. We have our platform, which is the RevMod platform, which is a fully human platform of 28 billion sequences that we can bring together in multispecific antibodies. And our first concept, or our first lead candidate with this, is RHO-104. RHO-104 combines aspects of Eylea and Vabysmo into a single antibody therapeutic. It blocks multiple types of VEGF as well as Ang2. And we are moving this quickly into the clinic. All the targets are validated in terms of blocking the VEGF receptor 2 and Tie2 signaling. The regulatory path is clear as it's an antibody-based therapeutic. And we've met with the FDA with our pre-IND meeting. And the pharmacology and pharmacokinetics have been established. The risk of immunogenicity is low as the molecule lacks an FC and is in a Fab-type format that is very well tolerated within the eye. And manufacturing is ongoing with drug substance coming together and drug product. Toxicology, the pilot tox has been completed, and we expect to be in the clinic in 2026. The binding affinities are all sub-nanomolar, so picomolar for VEGF-A and Ang2. And the preclinical proof of concept data is robust. RHO-104 outperforms both Vabysmo and Eylea in the rat laser CNVM model, but more importantly, this translates into higher species, with the non-human primate data also being quite impressive, with the regression of laser-induced CNVMs being at 90% for clinically significant grade 3 and grade 4 lesions. But more telling is the fluorescein angiograms. The column on your right is RHO-104. The column on your left is control. You can see the regression of the fluorescein images from pre and post, and the middle column is faricimab. OCT images are surprisingly human for non-human primates. But the control is to the left, and we see CNVMs, and for the clinicians in the audience, we see these kind of images daily with subretinal, intraretinal fluid. The middle column is faricimab-treated non-human primates with laser-induced CNVMs. We can see regression. And the final column is RHO-104, and what we see is regression of intraretinal fluid, this reduction in size of the pigment epithelial detachment, and real control. We're very happy when we see this kind of response after a single dose in the clinic. Repeat NHP pilot tox dosing, RHO-104 is well tolerated up to a human equivalent dose of 6 milligrams, which is coincidentally what the dose of faricimab is. No issues with intraocular pressures and no significant IOI. The safety was comparable to what was seen with Lucentis in the NHP repeat dosing. The clinical plan, we are planning to enter the clinic with a MAD, multiple ascending dose trial. This is a typical 3+3 design, 9 patients to 18 patients, depending on any dose-limiting toxicities, and with the 2 highest doses entering into a dose finding with a comparator arm of Vabysmo. 3 doses will be given at day 1, day 28, and day 56, followed by a 4-month observation looking for signs of increasing disease activity in terms of increased subretinal fluid or loss of 10 letters of vision. Patients who make it through the 4-month period will be able to extend into a long-term extension. The team is established all within the group of ophthalmology. We have a great bench here and a great group of advisors. We're looking forward to entering the clinic in 2026 and reporting back to you at this conference next year. Thank you so much.